Welcome back to one more session of bioinformatics. Today we are going to talk about rasmol. First of all, what is rasmol? Rasmol is one of the popular 3D molecular graphics viewer. You can see proteins, molecules usually, they in the 3D structure, okay, and it makes us work perfectly with smaller molecules. This particular software was developed by Roger Sale, and it is open source software which is available at the openrasmol.org link. This software is widely used, it's simple to use for simple operations, and we can find that you have also an extra command line interface where you can control the various events or various conditions of the rasmol. Okay. Now, if you go to look into rasmol, as we mentioned earlier, it is a graphical software, molecular graphical software, and various biological macromolecular structures like the proteins are being viewed using rasmol. So, what you have to do is you need to take the PDB structures from the protein data bank, and these structures which are being stored in the protein data bank is being viewed using rasmol. So, even if the 3D structure of that is not being available in the data bank, sometimes you can be able to view it using rasmol. In most of the cases, you will also get a 3D view of the protein in the in the protein data bank also. But for various purposes, sometimes you need to take the 3D structure of a particular protein, and might be you'll need to visualize it in a different way. Sometimes the active site residues might be needed to be selectively displayed in a different way. All that has been possible with the help of a software called the rasmol. Now, it was originally developed by Roger Sale in 1990s, and historically it was an important tool for the molecular biologist, since the extremely optimized program allowed the software to be run modestly powerful even in the personal computers. And before rasmol, visualization softwares ran on graphics workstations that, due to their cost, were less accessible to the scholars. But now we can see that each and every researcher can easily install the software rasmol in their PCs, either in the Linux as well as in the Windows platform, and we can visualize the protein structures in a three-dimensional manner. So now let's just explore the program. So when we just explore rasmol in Google, we can see that you get a link of called rasmol.openrasmol. Now I am used to Bing search over here, so I'm just clicking and going on to the rasmol. So you can see that you have different links here. You can either install, now according to your PC you can install what is, now I'm doing a Windows installing here, and here when you install you will get a link called the rasmol Windows will be explored, and as a normal procedure you just have to install it in your system. So let's see how the software looks. So now when I looked, I have installed it and it has been installed as raswin. So when you open the software, you can see that you get a box similar to this. I have not registered it and I am opening it here, and you can see that along with rasmol, now you have to open something. You need to open the thing which you have to view under here. So I'm opening a file. Now I'm having a protein here already 2FUM, which is already been downloaded from the PDB database. So I'm opening it here. So this is how a protein looks when it is being downloaded and viewed using a rasmol function. Now this is why X-ray diffraction that this particular protein has been obtained, and that's what 2FUM dot X-ray diffraction it has been shown. And here I have different ways to display it. It can be like its wireframe, by backbone, display by sticks, displays by space fills, display by ball and stick, display by ribbons, okay, display by strands, that's in one way. So display by cartons, display by molecular surface, like that. Different ways of displaying them are there. Okay. Now let's explore the different color options of it. Now you have different options here. If you give monochromatic, means one single color will come, and then the next one is a CPK. CPK is a color option which was given by Corey and Pauling, and later on it was been modified by Culton, okay, and hence the name CPK. And here all the carbon atoms will appear to be grey in color, the hydrogen atoms will appear to be white, oxygen appears to be red, and nitrogen appears to be blue. And this is the standard format which is normally followed by most of the textbooks. And let's look at the other option. You have Shapley option over here, and it is based upon different amino acid sequences. Different colors are being given. Now all the residues, now the places where there are yellow color, it indicates that those regions are being made up of amino acids like cysteine and methionine. That is, different amino acids are being given different colors based upon their properties, okay. Now where you have the regions where you have bright red, it indicates there the aspartic acid and glutamic acid is there. It's not that evident here. And if you are having a pink color, okay, here is the one which you have here, it indicates that it is having tryptophan over there. And if you are having a dark blue over here, it indicates that the amino acids lysine and arginine are present over there. Now coming to the next option that we have is regarding the chain option. So depending upon various chains involved, they will be having different colors. And the next color option is the temperature. Based upon the anisotropic temperatures of the various atoms involved in this, you will have different colors. If a high temperature is being present over there for the atom, it will appear to be red in color, and if a lower temperature or if it's a cold one, it will appear to be blue in color. And you can see there are intermediate colors also in between that. Then coming to the structure, this is something of much importance. You can see that it mainly shows that the different colors have been given for different two-dimensional structures. If you go to get a magenta color, it indicates that there is a helix over there. If you get a yellow color, it indicates a beta sheet. A blue color indicates turns. Somewhere here you have a slight blue color. So based upon the color itself, you can identify what all type of two-dimensional structures are being present in that particular protein. And that's about the structure. And we have another one called the model over here. It's just another showing a model one. I guess this is clear to you now. Now we can see also the command line which has been present in rasmol. Okay, you can see that there's a command line also over there. See, on your taskbar itself, the command line will also come, and you can type into the commands. Now, for example, select, HELICES, helices. So they have selected the helices, and then I am giving that color red. So all the helices will color to be red. Like that you can give different. Now if I get select sheets, okay, they have been selected it, color YELLO, yellow. So all the sheets have been colored yellow. So like this you can identify, you can give different commands in the rasmol command line also. Now let me just give a random command, select arginine. Okay, there is error they are saying, I guess. Select. This didn't. This is an unrecognized command line, I guess. I'll show you what is it. One sec. Yeah. So let's continue. The command line was error was being shown earlier. So I gave now select. Let me just type in select. Instead of the full amino acid, you have to give the short form ARG. So the atom is being shown. Now color it to be, the spelling should be correct, COLOUR, color it to be as blue, BLUE. So all the arginine residues will be appear to be in blue color. Like this, you can give different command lines on what you have to do in the rasmol software. Now to explore the whole command lines and all the features of rasmol is not being possible in this session. So just note this site live this web page which you have, where all the details of the rasmol software is being given. Now what are the internal parameters, all the section introduction, what are the examples. Now if you want to know about the bonds, how the bonds are being displayed, then if you want to find out different parameters, now if you want to find what is wireframe, like that, all the information regarding different features of rasmol is being given in this link. You can just note this, and the best way of learning is to explore rasmol with its features according to your need and the way how you need to depict different parts of the protein. You can also see that not only the proteins, you can also find the three-dimensional structures of nucleic acids also. So kindly explore and learn more. Thank you.